What we wanted to talk about, and this is really a joint project with Bob Weiss, so I put him up here on the author line to talk about this large kindred of FSHD patients here in Utah, and really talk a little bit about how the fruits of 80 years of engagement of this family in FSHD research is starting to bear fruit in some interesting ways. So I made a quick objective slides, but it's nothing to say we'll introduce you to the family and then talk about a genetic modifier study we're doing that relates to the family now. And this is maybe the start of the story, but it starts well halfway through. And this is a paper published in 1950 by Frank Tyler and Fayette Stevens that describes manifestations of FSHD in a large Utah family. And this is the family, and this is a pedigree that maybe some people have seen. There's over 1,200 people on this pedigree, and over 150 of those affected with FSHD. And really, even with this size of the pedigree, we're only seeing a part of that story because there's lines that were lost to follow-up that aren't even included here. But I'm going to just walk through this study from Tyler and Stevens, and then talk a little bit about how this family's been engaged with us in FSHD research over the years, and where we're headed with that. And to start with, I pulled these pictures right out of the original study, and what I think you can see is that this patient obviously had FSHD. So I pulled this summary also, and this is a lot of words in this next little bit, but pulled directly off the summary of Tyler and Stevens from their study. So they describe this study of 1,200 individuals in a single kindred with FSHD, and they make a clinical description, and they're very precise about the clinical description, and they highlight the pattern of muscle atrophy in the facial and pelvic and pectoral girdles and other muscles. And you can see that in the photos, right? You see the horizontal clavicles here, and the sort of sloping shoulders, and the scapulae that's overriding here in this patient on the top. And then I think this really prominent atrophy of the humeral muscles, so biceps and triceps. If you look at the lower arm, he's got more atrophy up in the upper arm than the lower arm. Anyone looking at these today would say that this is FSHD just looking at those two pictures, and the picture that's described in the paper in these patients is very typical. They did have this interesting comment that the disease is usually benign, and only an occasional patient being severely handicapped. I think people might argue about how benign it is, but I thought that was just an interesting comment from the authors back then. And the other important thing they describe is the inheritance, and what they describe is a Mendelian dominant pattern of inheritance with complete penetrance and variability in the severity, or we'll call it variable expressivity. And that's a really characteristic feature of FSHD, but they noticed it from the very start, and we'll talk a little bit more about that. And then this fourth point is kind of interesting and never caught my eye till I was looking at it yesterday, but they say the occurrence of other diseases and traits has been studied in detail, but no significant correlation. And so these are things that were available to these guys to study in 1950. So they looked at blood types, for example, and does the inheritance of FSHD correlate with the blood type, or other physical features? And what they were trying to do is genetic mapping. And the markers they had were phenotypic markers, so disease markers or blood type markers, but they didn't have the molecular tools that were available years later that helped identify the D4Z4 locus and lots of other genes. But I think that's interesting that they were already thinking about this way back then, that there must be a way to figure out what chromosome is this disease localized to, and how would we find it. So this is another quote from the paper that really highlights the severity, the variability and severity of FSHD, which is really a hallmark. And they noticed it right from the start, but they say it's clear from the data that the severity in the parent has little influence on the severity in the children. And they observed severe cases in the offspring of parents with mild dystrophy, and then the opposite, they also found certain of the most affected individuals could have milder signs. So that variability has been a part of this and that story of variability from the very start. So I'm going to just give you a quick primer on genetics of FSHD. Many of you probably know these things well enough, but just so that we're all on the same page about genetics. So FSHD is inherited in a dominant fashion. That means you only need the mutation on one of the two copies that you have of every gene to have this disease. And that means also that the mutation passes to 50% of the offspring. And the locus that's important in FSHD is this locus called D4Z4, and I'm intentionally not calling it a gene. It's really just a spot. And I put a little figure up here. It's way out on the end of chromosome 4, right near the very, very end. And D4Z4 is not a gene per se. It's a macrosatellite repeat. And a macrosatellite is just a repeat. It's about 3.3 thousand, so about 3,300 letters long. And you have multiple copies of that repeat out there at the end of chromosome 4. And a normal person has anywhere between 11 and 100 copies that are represented here by these triangles, right? And if you have more than 11 copies, you're normal, you don't have FSHD. And if you have less than 10 or less copies, then you have FSHD. And there's some complexity to that, and you have to have that on the right genetic background, and we're not going to spend a lot of time going into that. But the basic idea is that if you contract the number of those repeats, you have FSHD. And it's been known for a long time that the severity is loosely correlated to the number of repeats. So the lower the number of repeats, the more severe the disease, but it's not a perfect correlation. And then the gene that we're interested in FSHD is a gene called DUX4. And if you think about each of these repeats, that's about 3,300 letters long, there's a tiny little gene in there, and it's not even a full copy of the gene, but the gene is called DUX4. And that gene is an interesting gene. It regulates how other genes are turned on and off. And in normal humans, it's turned on when you're a 4-cell embryo. And it's on for a period of hours, and then it turns off forever. And what we think is happening in FSHD is that gene gets turned on at a time when it's not supposed to be turned on. And there's all kinds of research and stuff to think about how that might cause disease. And we're not going to talk about the rest of it, other than just to say we're interested in the genetics of this down the road. So you need to know what DUX4 is and what D4Z4 is and what the repeat numbers are. Now, I'm just going to give you kind of a history of Utah and a history of FSHD in Utah at the same time. And you notice that they kind of started at the same time. So in 1847, Mormon pioneers came into the Salt Lake Valley and settled here permanently. And about 2 years later, a man named John Parker and his family immigrated with other Mormon pioneers into the Salt Lake Valley. So that's 1849, 1852, as different parts of the family came here. And John Parker had FSHD. And they lived here. They moved to different parts of the state, and sometime probably in the late 20s, early 30s, a orthopedic surgeon named Samuel Baldwin came to know some of these patients with FSHD and became interested in the families. And he had collected a number of families, and then in 1936 passed them off to a man named Fayette Stevens, who was a geneticist that arrived in Utah about 10 years before, and mostly was interested in plant genetics, but came across a number of different pedigrees of human disease. And his first kindred was a study of osteogenesis imperfecta. That's kindred 1. And the second family worked on is the Parker family with FSHD. That's kindred 2. And then he studied a whole bunch of other families. So cancer genetics, so the breast cancer genes were first discovered in Utah families out of work first started by Fayette Stevens, colon cancer genes, a number of other genes. So this blossoming of genetics here in Utah started almost from the very start with FSHD. And as a part of those studies, the very first NIH grant, so when we think of our main funding mechanisms right now, they're looking for grants from the NIH, and the very first NIH grant to go outside of the NIH's internal research was made to investigators here at the University of Utah to study the genetics of muscular dystrophy. That was in 1946. That funding and that additional work they were able to do led to the paper that we've been talking about, 1950, Tyler and Stevens' first publication on this family with FSHD. And they carried on work with that family over about the next 20 years. So we have some notes from them even through the late 60s of encounters with this family that had been following closely for a long time. And then there's a little bit of a gap, and Mark Leppard in the 1980s picked it up at that point and is now collecting families with different genetic disorders, but now with a number of tools that had just been developed, and those were largely developed here in Utah, but to do genetic mapping. And so they were interested in all these families that had been known for a long time. And he started recruiting members of the family and called them kindred 1462. So we talk a lot about 1462. But not only did he collect phenotype from the family, he collected DNA samples. And so those DNA samples still exist from Dr. Leppard's lab, and we have them. Around the same time, Stephen Jacobson and Daniel Prezer, the founders of the Society, of the FSHD Society, started collecting samples from these families. And ultimately, these samples developed cell lines and other reagents that are available even still through Coriell. So some of those original reagents that are available to investigators all over the world come from family 1462. A few years later, Kevin Flanagan, who was here at Utah at the time, picked up some interest in studying FSHD, and his work with Dr. Leppard started now doing some genetic analysis on those DNAs that had been collected over several years. And then Dr. Flanagan went to Ohio State, where he works on gene therapy and does some really good work there, but we had a little bit of a gap again. Kevin's main publication was to talk about that within the same family, patients always have the same number of repeats of that Z4 gene. And I'll show you some data from that paper in a little bit. But that was 2001. And then what happened after 2001 is a real revolution in genetic technology. And we put a marker here at 2007 for the GWAS era beginning. And GWAS is just a technique that we can use to map genes, and particularly for complex sort of traits. But there's a number of other important genetic advancements, including next generation sequencer, ability to sequence the whole genome all at once. So these things all happened after we had a little bit of a gap in our study for the family. And that leads us up to the more recent thing, which is a 2018, Dr. Weiss and myself and Dr. Johnson started a study to try to understand genetic modifiers of FSHD. And that's funded through the Wellstone program in collaboration with University of Massachusetts Wellstone Center. And it's an active project. So we'll talk a little bit about that project. This is just to give you a sense that these are real people who worked on this project over many, many years. This is Samuel Baldwin, a photo from his obituary in 1944. And it turns out this was front page news in the Salt Lake Tribune in October 1944 on the top half, if we think about the way we used to think about newspaper articles. So it was the most important story of the day that this physician had passed away. This is Fayette Stevens. If you'll notice in the background here, he's doing some teaching with these students about this kindred to this FSHD pedigree. Frank Tyler here in the 1970s, he took a more prominent role in the metabolism department. And then here, Kevin Flanagan and Mark Leppard, who've worked on this project for a number of years. So these are real people across now about 80 years worth of engagement in this study of the kindred 1462. So where do we come in? Where do I come in? And this story starts with a mysterious box appearing on my desk one day about 5 years ago. And that box had notebooks in it. And I opened the notebook, and this is the first page of what was in there. And it didn't take me very long to realize that these are the original Tyler and Stevens case report forms. And what this did is unlock some things we didn't know before. So we had known about this pedigree for a long time. We'd seen it. It was republished in Dr. Flanagan's paper in 2001. And what we didn't know is who was who. Who was this person? We kind of knew because that's John Parker, the founder. But after that, who was any one of these individuals? And one day we made a bigger picture of this. So I'm going to zoom out on the middle part of this pedigree and show you a couple of things. So this is generation 1, individual number 1. And you can go here, generation 2. So this person had 10 children, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Right, so this is John Parker Sr. And now I can see the numbers that tag him. But what we saw in those notebooks is that this is generation 1, individual 1, John Parker, born in 1775. And it lists his 10 children with their names and birth dates. So what this mysterious box of folders did for us is it opened the key to understanding who were the exact individuals on those pedigrees with 1,269 people, and what was their phenotype assessment and when was their onset. So you can look through here. Individual 6 is John Parker Jr. We see his multiple wives and the individuals affected from that family. And John Parker here obviously noted as affected and Mary Parker, which become the 2 most important lines that we're following. And I'll show you a little bit more about that in subsequent slides. But it's sort of the Rosetta Stone of Kindred 1462 that allowed us to really start putting together an individual and a pedigree and then tying that now to people who are still here. So this is our redrawing of the original kindred. This is John Parker Sr. And I think you can tell even just by looking at his image that he probably has FSHD just by the muscle atrophy in his facial muscles. There's an interesting little comment we found in one of their pioneer histories that they had a spring carriage, which they made some adaptation to get him across the plains. So he essentially came here in a wagon from Iowa in 1849, but wasn't able to walk that whole thing. They took him in the carriage. His son, John Parker Jr. is that individual number 6 that I was showing you in the last pedigree, and Mary Huskin Parker. And those are the 2 founders from which we have most of the data now. And then these are their children here, Alice Parker, William Parker, and others. And we've kind of coded them into separate family groups here by different colors. I'll show you a little bit of things we've learned about each of those. And just an interesting historical note, Mary Huskin Parker is actually a really prominent historian in the sense of Mormon pioneer history and understanding of what those tracks were like. She's a prolific writer. And so we have a book of her journals that have been collected from her time crossing the plains. And it's very interesting that she's not, and this happened in her early 20s, but not very descriptive about her muscular dystrophy. She didn't describe it to me. I think you can see from her picture as well that she's likely affected just even to look at her picture. So I'm gonna go through 3 different families and a couple of things we've been able to observe by just collecting the historical records. And then the first comes out of Mary Huskin Parker. So that's her right here at the top. And I'm going to show you some things about these people kind of a little further down. So I highlighted her here in red. This is where she fits in that original pedigree, what we're seeing here in the pink one. And this is a family which we don't have a lot of information right now. So these little pedigrees I'm going to show you that are drawn out. The red one is the proband of that family. And these are people that we know. And if there's a hashtag by it, that means we have a DNA sample. So now we've got a couple DNA samples from this family. And this individual is still living, 93 years old, and started noticing symptoms in their 20s. And they were actually examined by Dr. Tyler in their 20s. That's him here. He's on the original pedigree. And noticed just some facial asymmetry from the start. And he worked until he was 61 years old and lived in a wheelchair since age 70. Seen briefly by Dr. Flanagan back in those early 2000s when he was doing his work here, but then lost a follow-up there. And then it's kind of interesting that his grandson had been diagnosed a few years ago at age 30 after 2 years of just worsening back and shoulder pain. And he'd been unaware at the time that he could have the same disorder that was in his grandfather. So there was some loss of recognition of this over the years in some families. And then soon after his diagnosis, his mother was diagnosed as well, and she had, symptoms that dated back to her 20s or 30s when that diagnosis was made. So that's just the first family we'll highlight. And here's the second one. So it's just different edge of the pedigree here. And interestingly, this is the family 33 in Jones's paper that describes the cell lines. And here they are on the original pedigree up in a corner. And what you see is that there's just one affected person, so a lot of unaffecteds, and then nothing below. But here's where we start below. So this individual 2-5298 is this one. It's the last one in the pedigree, but now we know a lot of other individuals in the family, and I'll just show you some of their this is that original record, generation 4, number 53. So that's this person's father, described by Dr. Baldwin and Dr. Tyler as having FSHD that was severe. And then we can see the family, and only one of those individuals was noted as having the trait. And that's this one. I'm going to just show you that was him. That's him right here. But siblings not recognized to have the trait. And what we see now is that this individual 298 is actually this one. So this person was thought to be unaffected. And this is that person's son now. So 298 son, this is now generation 6, number 396, and had symptoms that started pretty young. So age 12 or 13 with some difficulty in push-ups and facial weakness that was noted while playing instruments. It became more noticeable in the 20s and 30s. So this isn't all that unusual description for FSHD. And his son now age 26 with symptoms arising in young adulthood. So now third family, just to give you a little bit of information on, is descended from Alice Parker. This is her family here, the ones I'm going to show you. And again, these are going to be a lot of individuals that initially marked as unaffected in the original pedigree. And we have this pedigree a little more filled out with people that we know. And so this one, number 205, I'm going to tell you a little bit about, and then his two sisters. So this is their father, George Isom, born in 1875. And it's seen by Dr. Baldwin, that original orthopedic surgeon, and seen by Dr. Tyler, who both thought they had severe muscular dystrophy. And their youngest, and his youngest son, Wayne, who was born in 1922, noted to have also severe muscular dystrophy, and we can see those original notes. But also a couple of things we noticed on here is, one, that they have records, and we don't actually have these records, but there's a research file that tracked this family up until the mid-1960s. This is an original note from Dr. Tyler's visit in 1947. So we have copies of those notes, and it describes his onset in his early teenage years. And a second visit in 1974, this is with Dr. Zeider, who took over for Dr. Tyler, and it gives that same description of his onset in his early teenage years and progression across his lifetime. By this time, he was well into getting into his 50s. This is just his handwritten intake note that describes his onset as well. Started to become limited in my teens, he says. So we have a pretty good sense for the phenotype and its severity in this individual across about a 70-year timeline from onset. Now, his sister Alice is a twin. I'm going to show you where she falls in that original pedigree. She is individual number 200. So this is her here, and her family was also seen some children that were probably too young to phenotype and two older ones that were thought to be unaffected. And sort of interesting finding we noticed as we got deep into these records is that she was originally marked as unaffected. She has a secondary annotation that was done in the 1950s that marks her as affected. So these records actually allow us to establish in this individual an onset somewhere between her original visit in late 1940s and then her second visit in the late 1950s, which would have been in her 40-year-old kind of time frame. And that allows us to sort of contrast her with her brother who had an earlier onset and kind of demonstrates the variability in severity. Her sister La Prielle also thought to be unaffected on that first visit and re-examined and later noted to be affected. So it's kind of a treasure trove of information coming out of these old documents. And we've spent a lot of the last year trying to collect all this older information and pull it together into one resource that becomes usable now, and with computerized records and all that. And that kind of leads us now to the genetic modifier project and really trying to get at a better understanding of the severity of FSHD. That severity we talked about a little bit, and it's explained in part by the number of D4, Z4 repeats. So if you remember, the lower number of the disease is more severe, but it doesn't explain all of it. And even as that, two siblings I showed you, it can be a variable severity even in the same family. Well, we know they have the same copy number for the D4Z4 repeat. And we want to know what's controlling that. So can we find some other gene or some other environmental factor that's controlling the variability to FSHD? And the answer is we now have the tools to do that. And we can do it, but we need a lot of patience, and it helps if they all have the same mutation, right? So we're not fighting the variability from the mutation itself. So Kindred 1462 is really a unique resource to answer these questions about severity and progression and variability between individuals. And for us, we now have this multi-generational family resource that's been collected now over 70 or 80 years worth of involvement. And we have DNA that had been collected even back into the 1980s. And I think our oldest DNA was for an individual born in the late 1800s. And it's just an irreplaceable resource. And now with genetic tools that didn't exist in those days to do the analysis. So this is really an important resource, we think, for the FSHD community to pull all of these records together, pull the DNA samples together, and then be able to do more modern genetic studies. And the goal of that really is to understand severity and understand variability. And if we understand the genes that control severity to the disease, we can target therapies at those mechanisms that are controlling that severity in hopes that we can make everybody a milder case. So that's really the goal of the Modifier project. I'm going to show you just a little bit of data that's been collected over the years with the thought about modifiers. And the first bit of data was collected by Kevin Flanagan in his 2001 paper. And this is just a sort of pared down pedigree across the whole thing. And what we did is we highlighted in yellow across this pedigree each of those individuals. And one thing you notice, they come from a really broad spectrum of pedigree. So this person G is really not all that highly related to number F, right? They're pretty far apart genetically, but they still carry the same mutation. And what Dr. Flanagan showed is that they all have a 6-unit D4Z4 contraction. So every one of those people across that whole pedigree. So that really establishes the stability of the primary mutation. And that's a really important advance that we started with a few years ago from Dr. Flanagan. This is just a sampling of people that we've collected so far. So we have about 224 DNAs in Kindred 1462. 13 of those are traced back to the original pedigree. Some of those patients are no longer with us and were samples collected back as early as the 1980s. And so we've started a bit of genetic analysis on those, and we can trace back mapping for the FSHD locus, and now working out of that to try to collect some new patients. So what are we doing next? Probably one of the biggest things that we're doing right now is digitization and analysis of those existing records. How do we make that searchable and analyzable, and then link the DNAs that we have into that pedigree? And one of the things we did is just development of a haplotype assay. So this is a really easy screen, and FSHD testing in the clinic is not simple at all. And if you've had testing, know this, that you need a fresh blood sample, and it goes out to a special lab and does a southern blot, which is a really difficult technique to perfect. But for this family in particular, we've developed a number of a small set of genetic markers that we can just easily screen for individuals who carry that same chromosome that was inherited from John Parker, the original founder. So that is an important advance that lets us just rapidly screen through people. And then our next steps are really in identifying new members of the kindred. And there's a couple of other larger kindreds here in Utah, and we think that may spread now out from Utah. And one of the things that made this possible in Utah is that people stay put and they know their genealogies. But in the last few years, people move around a lot more. So these families are becoming more dispersed. But the goal is to collect a DNA sample, both from affected individuals and unaffected individuals. And it turns out for the type of genetic analysis we're interested in, it's really important to have the unaffected people too. So we're collecting DNA samples and phenotype assessments from people. And the phenotype assessments are really driven to an understanding of onset, severity, and progression, and understanding who is a more severe and who's a less severe case. During COVID, that's been really difficult, and we're doing that a little bit by survey and phone interviews. Post-COVID, and we had even planned this for this summer, but doing in-person evaluations with families, attending some family reunions and getting to know people a little bit that way. In the future, one of our important goals is to actually secure funding to keep a longitudinal study for these more discrete phenotype assessments that we can do in person. 'Cause there's only so much you can learn from those historical records about things that we're interested in. So that's one of the more important things we're interested in down the line as we collect the family, collect the historical record, and bring all of that together into one resource for the community. Last thing I'll just tell you about is part of that recruitment effort, and this is Roots Tech Conference. It's the largest genealogy conference in the world that happens to be here in Salt Lake City most every year, and it happened late February, so just before COVID turned the world upside down. So we had a booth there at the Roots Tech Conference, thinking that there's a good chance descendants of John Parker would be there, and give us a chance to just do some outreach for the community, talk about muscular dystrophy, talk about genetics, and hopefully identify members of the family. And we did. We were able to identify several descendants of the FSHD founder, including this one as the most important one. And this is Beverly Dowling. I'm going to move my thing that's in the way. And an individual stopped by the booth. He says, "I think my mother may know something about this family." And it turns out she's a member of the family. I think this is her down in here somewhere, if you can see my pointer. But she's actually a descendant of John Parker and a member of the family, but in an unaffected line. But more importantly, Beverly is the person who drew that pedigree. And so she worked with Dr. Stevens and Dr. Tyler back in the late 40s. And she was the go-to person with her hand-drawn amazing pedigrees. And it was such a neat experience to sit and visit with Beverly for a few minutes and pick her brain about how they were thinking about the genetics back then and see how they were doing that project, and actually to be a participant across multiple generations with her on the same project. That was a highlight of that recruitment effort in the Roots Tech. And so COVID hit a couple weeks later. We haven't been able to talk to her again because we don't want to try to get in the way of her living in 10,000 more years, so we can talk to her again. But so that's been a really neat project to work on that really crosses generations. And that work has been done by a lot of people. There's a large group here at the university with Bob Weiss, who's one of the principal investigators with that, and Diane, who does a lot of the molecular work in the lab, and Britt, who's been collecting these records and drawing the pedigrees and making them work, and Sarah, who works to get people enrolled. And a special shout out to Kathy Gilmore, who's actually a neighbor who developed an indexing tool for genealogical records. And this has been a really key resource for us to pull together those old files. And obviously we've been working with Dr. Flanagan for many years on this as well as the group at the Wellstone FSHD Research Center, who's a part of the funding. And then I just listed there the legacy investigators, which really go back to for a long time here in Utah. So I'm happy to take questions, talk about the study in any more detail. I put a little QR code here if you're interested in participating in our studies, or if you think you might be related to family, there's some instructions how you might do that, or you can just email us. But that's all I have. So.
Oh, wonderful. Well, thank you so much. That was just such an interesting story, blending history and science and medicine and bringing it up to the present day. So thank you for that. And thank you for your whole team for working so diligently. I know that a few years ago, we at the Society were really worried that the study would just kind of be collecting dust, after Dr. Flanagan left. And so we're thrilled that you came and picked up the
Yeah, it was an interesting series of events because Dr. Flanagan left just at the start of my neuromuscular fellowship, and we did drop a few things at the time. Had some time now to pick them back up again.
That's so great. So I just wanted to quickly mention to our listeners our newest issue of the FSHD Advocate. You'll notice the cover. The theme is we're keeping our focus. The cover is a scene of, I think, Bryce Canyon in Utah, and with like an iPhone camera, like focusing on the image. It's a subtle tribute to this study because we have an article in there about the amazing origins of FSHD research in America. So, so when you get that and you're wondering, like, why did we put Utah on the cover? There's Bryce Canyon on the cover of the magazine. That's why. And in that article, we also included these web addresses and the QR code, which if you have a smartphone, all you have to do is point your camera at the QR code and it'll bring up the web address, and you can just follow the instructions to see if, you know, if you might be a family member or if you'd like to volunteer for the study. So I have a question from Ellen. She was wondering, do you know who sent you that box that mysteriously landed on your desk?
We're trying to track down the origin of the box, and it's been an interesting journey to figure that out. We're pretty sure that Kevin had checked that box out of, Dr. Flanagan had checked that box out of the archive back in the late 90s when he was doing his study. And I think it was passed down from there to Jacinda Sampson, who worked with him, and from Jacinda to me. So I think from Jacinda, it just got left in an office and then just handed off by chance. So what we found out more recently is that that box was in a set of records that was in an archive placed there in 1991 and set for destruction on a 25-year timeline. And so a phone call was made to the Department of Human Genetics in 2018 asking if we still needed the records, and the answer was no, and they were all destroyed except for this one box which had been checked out in 1995 and never returned. So the box was saved. It turns out there was a kindred 2 folders in there, but there was also a kindred 66. So there's a second large kindred here in Utah of FSHD that was studied but not published by Tyler and Stevens. And that family had also origins in early Mormon history, but had come to the United States back in the 1600s, so about 200 years prior, and carrying FSHD out of England again. They also have a 6-unit repeat, so we think they're a good second resource to study for the modifiers as we collect some members of that family as well. And turns out there's 5 notebooks in the box, and we just found a 6th notebook, which is the second half of that kindred 66 family that we didn't even know the notebook existed till Dr. Flanagan found it in his files a couple of weeks ago. We have that now on my desk.
Wow. So lucky that box wasn't put through the shredder. Oh my goodness. Well, all right. So we had a question, a vocabulary question. People want to know what does phenotype mean? So that is a somewhat technical term.
Oh yeah, I should probably have explained that. Phenotype is just the clinical manifestations of the disease, right? So if we're talking about hair color, phenotype is blonde or brunette or gray, right? If we're talking about eye color, it's blue or brown or green. So in this case, it's FSHD is the broader phenotype. One important question we have about genetic modifiers is what is the phenotype of the modifier? What is the modifier changing? And so then we try to think more deeply about FSHD and the drivers of FSHD as the biology and things. So we can use phenotype in a really more specific sense or in a more broad sense, but basically just means the manifestations of the disorder.
Great. I was curious, have you or anyone tried to identify relatives of this family that were left behind in England?
We've had an interest in that, and I've talked to Volker Straub a couple of times about it, but not been able to nail it down yet. They actually have a really nice registry in England of FSHD patients, including DNA samples. So the little molecular tags that we identified would be easy to screen in those families. And so that's one of the things we definitely want to do is screen that back into England, because we think it's probably preceded John Parker's departure from there, right? It's probably there.
Well, that would be really cool to find.
It would be really cool to track it back,
So we have a question from Shireen that she notes that many people seem to develop symptoms in adolescence. You mentioned that in some of these case reports. So what is the role of hormones? Do you have any insight into the role of hormones into the development?
Yeah, I probably don't have a lot specific to hormones, but it's been an interest, and there's been some investigation out of a group in the UK about gendered effects on the transmission. So I think like a geneticist, I guess. So I think about transmission of genes. And if you transmit that gene from the mother to a child versus the father to a child, does that make a difference? Is there a hormonal connection to that? Those are things we don't know. This particular kindred is a really important way, I think, to identify those effects in transmission. But I don't know a lot about the hormonal influences and what drives, that onset and as it correlates to puberty or things like that. I think it's a really interesting question, but not one I know very much about right now.
A question about epigenetic modifications, and I have a similar interest. Have you analyzed the methylation level of the D4Z4 repeats across multiple individuals, both across the family, but also, across this kindred, but also over time, if you have samples that were collected, decades ago and are able to recollect from the same individuals. I don't know if you've been able to do that.
So this is an area of interest that we don't know the answers to, but FSHD is really a very interesting disorder in its connection to epigenetic controls of gene transcription and gene activation. One of the things that we're interested in studying now, in particular with this kindred, we have DNA samples in some cases that were collected in the 80s, maybe a second sample that was collected a decade later, and now a sample that's collected now. So we, in some cases, have these 3 different time points where we have a DNA sample and can look at methylation. So we think that's a pretty interesting way to think about modifiers of severity. Does the methylation change over time, and what's controlling those changes? Or is it just stable? Is it stable across time and across its inheritance? One other thing we don't know is it stably inherited? So we know that the length of the D4Z4 is stable, but we don't know is the methylation levels stable between generations. And certainly we know that the FSHD2 genes can alter and control a little bit that methylation status. So is there some modifier effect from those genes? Those are all things that we will study as a part of this project. We're now developing a tool that will let us measure methylation in a really precise way using a technology called nanopore sequencing. We're hoping to get some additional funding for that work, but that'll really open the door to studying those things and look at the methylation across time.
Yeah, great, thank you. There's a question if the research so far has come across candidate genetic modifiers.
Not yet. That's sort of our next step as we get enough individuals into the database and get DNA and phenotyping information from them all. That's really the next step, but we don't have a candidate modifier as yet. We have a couple areas we're interested in looking right up front, and these come out of DMD studies that we've done and others for modifiers of Duchenne muscular dystrophy. So there'll be some sort of easy picking at those genes to do analysis there, but then looking broadly across all the genes. And that just takes a bigger cohort than we have access to right now, and getting the phenotype data a little more fully collected. So that's really the next step that we'll be doing.
There was a question, how many other families are you able to study? Are you pretty much focused on this pedigree is pretty extensive, so it's like, but
yeah, we're interested in anyone, and the way the progression of the study will go is that we'll hopefully find a number of candidate modifiers, and we'll need other families to study them in, right? We don't want to find something that's exclusive to this one kindred, but be able to take it out from there. So actually identifying people that are interested in genetic studies is really important as well. And it'll be the next step as we get to a set of candidate genes, and then doing validation studies to see them in other populations. And we are interested in obviously the other line, that kindred 66 line is last name Garner. We're interested in collecting that family because we know we have some access to it. There's a couple other probably lines here in Utah that Dr. Flanagan had ascertained before too. And we do think some of those families may have dispersed across the country over time. We know that the Garner line, which is kindred 66, didn't all come west with Mormon pioneers. They originate out of Virginia and North Carolina area when they came in the 1600s, but then stayed largely in that area until their move with Mormon pioneers into Iowa, where a large group stayed, and then into Southern California. So there's been a large dispersion over the years too.
Okay.
So you could connect to one of these, right? People, as particularly, you're a six repeat size.
Yeah. There was a question that just, oh, okay, was answered, but it's worth repeating. Someone said, what do you need from us Garners? So there must be a family member of the Garners.
On the line with us. Click on that link, that QR code, or email us, and we'll get you on the study.
Okay.
And I guess to reiterate one point I think I made earlier is that actually getting whole SIBShips, so a whole family affected and unaffected kids is really helpful. And there's maybe a little bit of perception that those unaffected individuals aren't helpful, but they really help us sort through some of the genetics that to have the person who doesn't have it as well.
Right. So there was a question before asking what is a SIBShip? So that's all of the siblings in a family, both affected and unaffected. Yes. Okay. And let's see. Oh, someone was wondering if all the, I guess, current living members of this pedigree, are they aware of this history and the story?
Part of the reason we're talking is to get this story out there, and part of the reason we're putting it in the newsletter is to get the story out there. I think, and that first family I highlighted is one where they had just lost track, right?
Mm-hmm.
So they had been engaged with Dr. Tyler and Dr. Stevens back in the 40s, but then kind of lost track of it, and it got picked up from a grandson that didn't know about that whole history, didn't know they were connected to this larger kindred. So, and we think there's others out there, and certainly there's at least one family that Tyler and Stevens never tracked down. And so we don't know much about that one right now.
Okay. Let's see, there's a question from someone who says, in her experience in meeting people with FSHD, there are often multiple other disorders present. I don't know if you have observed that or if there's some correlation between their having FSHD and having other conditions.
Not that we've come across right now, but it would be an area of interest. Yeah. Sometimes that just comes down to asking the right questions, right? Is there anything else going on? Is there some other, I know a lot of patients have pain and other related syndromes that tie to that are sort of indirectly to the dystrophy itself, but common. Obviously there's connections to hearing and visual issues, particularly in the younger people that are known, but we haven't come across other things.
Yeah, it's interesting. I have a gut feeling that on some of the very severe early onset, infantile onset FSHD, that there are other complications like GI issues and so on that may be a developmental issue. It's kind of or something. But
and this probably goes back to the role of that gene as a developmental gene, right? So the timing at which that gets turned on may play a role there.
Yeah, I was thinking. And in other cases, I've met people who have both FSHD, and it seems so unfair. They also have multiple sclerosis or also have diabetes or some other, and it's just FSHD doesn't necessarily protect you from getting anything else, but whether it's more prevalent.
And we have seen stuff like that in our other dystrophies too, or a patient will have diabetes or have something else like that. It doesn't necessarily seem to be connected, but it's hard to know.
Right. So there's a question, you mentioned hearing loss. Is there any insight into how the DUX4 gene might contribute to hearing loss in FSHD patients?
I don't know the answer to that one. I'll punt.
Yeah, okay.
There's a lot of research. I had another presentation I did where I just put charts with number of publications, right? So it's a chart that looks like an exponential curve, and it has number of publications that mention FSHD and the number that mention DUX4, and they're both pretty interesting exponential curves. So these are things people are all interested in. A lot of research going on about that stuff.
Okay. There's a question asking if it's possible to predict severity of symptoms based on the number of repeats. You kind of addressed that a little bit in your talk, but if you can talk about it a little more. I think every patient wants to know what their prognosis is so they can.
Yeah, this would be a really important thing. And I think as we come to understand maybe what some of these genetic modifiers are, we could start to build maybe some predictive models around those. Right now, we just don't have what are the parameters at which you would make that prediction beyond just the contraction itself, and it doesn't seem to explain enough.
Right. Comment here from Alexandra Belayu. It's a very distinguished science in the field of FSHD. She says, congratulations for the very nice story. Couldn't use a simple hypothesis that it's DUX4 expression level that may define phenotype severity, or maybe DUX4 target gene expression level. And she suggests that this could be studied in the cell lines established from the blood cells collected from this pedigree.
Yeah.
What are your thoughts about that?
Yeah, those are all important things to think about, right? There's a lot of ways to think about modifiers of severity, and one can just be coming right out of the gene itself, right? But then the next question is, what's controlling the DUX4 expression? What are the drivers? And maybe they're not all genetic. Maybe some of those are environmental modifiers too. And so there's a lot to learn there.
Okay. There's a question here, more of a clinically oriented question. Can the child of a parent with FSHD, and the child says child does not have FSHD, can that child pass it on to their children?
Yeah, that's an interesting question. So if we know that the child doesn't have the mutation, it would probably not be very likely to pass it to their children. Thinking like from a affected parent to an unaffected child to a next generation. The question there is an interesting one that we'll try to approach with the study here of penetrance. And there's always been a question about non-penetrance. And what that means is that maybe you have the mutation, but you don't manifest disease. And there's a bunch of different ways you can think about that. From the original Tyler paper, they actually think there's 100% penetrance. So if you have the mutation, you always have the disease. But then I showed you some examples of individuals marked as unaffected in the pedigree that may have just not had the disorder for a long time. Mm-hmm. And in fact, that one individual, Alice Isom, didn't become affected until her 40s. So then it's just a question of, are you pre-symptomatic? And that's a testable thing. So we can test that with genetic testing and answer the question. And so then you would just know the answer.
Okay. So one more question for now, I think. Is there a role for AI or machine learning in the search for modifiers? So as you start to accumulate big data sets, how are you going to analyze that?
Yeah, I think there's a role for these, algorithms that can see things that a person can't see just by looking. We don't have a lot of experience with AI, and genetic analyses are rapidly getting better and better. And so as those things evolve around, as AI evolves around genetics, I think those would be tools we'd be interested in using over time, but not that we don't have that sort of skill set right now in our lab to do it.
Oh, there was actually a few very interesting questions in the chat area that I didn't see until just now. There's someone who's saying, I am part of this family line and have several in my family that don't want to believe they have FSHD. What is the best way to talk to them about this? Would there be a way to have our whole family tested? We were tested in the 1980s, but never heard anything back since then.
Yeah, I wish that we didn't lose track of things like that. There are ways to just get together with the whole, and one of the things we're really interested in is actually just engaging with people who may have been, seen by Dr. Leppert and his group back in the 80s, or Dr. Flanagan after that, and just making some connections again. I know there's some families out there that had met with Dr. Flanagan, and he was doing some diagnostic testing even back then, but didn't always get results back to people. And we're interested in rectifying those things. For people that are a part of these larger kindreds, we can actually do the testing pretty simply, just off a DNA sample, and tell if you have that same chromosome. So it's a little simpler than if you were not connected to one of the original pedigrees. And that's just because we've developed the markers to test it simply. But if you just send us an email or something, we'd love to talk to you individually and get back to it and reconnect. The part about, family members not wanting to hear is a really hard problem, and maybe people on the line can better address that than I can, as not being someone who lives with this every day. It is interesting that it's in certain families kind of been lost to follow up because of those things. And we're really, I think there's been maybe a perception that there weren't things we could do. And so it may be frustrating to be into the doctors about it. But I think we're rapidly approaching a time where we can do things.
Yeah.
So getting people involved in the research is really helpful to move that whole thing forward.
Yeah. Well, now that so many people have become familiar with Zoom, it might be possible to get a group of family members together, even, fairly easily, just to talk about it. They don't need to say anything about whether they just to learn about it. And we'd be happy to help facilitate that too. So.
Yeah, and we have active society chapters here in Utah, and another one that's just starting up in Idaho that can help with that. And if it's outside of there, we're happy to set up conversations with families and have a discussion.